Starting off with the introduction, in recent years, a decline in pollinators has been reported worldwide. Western honeybees specifically make up 80% of insect pollinators and play an incredibly important role in pollinating a wide range of crops. Nutritional stress is one of the top driving factors behind colony loss, and thus, access to nutritious food sources is crucial for the success of honeybee colonies. A possible reason why bees have been struggling with their nutritional needs is because our agricultural landscape has become increasingly monocultural. In these ecosystems, that often means a deficit in both the variety and nutritional quality of the food that's available. This creates a misalignment in what bee colonies actually need and what can be provided by the environment. Pollen is the main source of protein for adult bees, and the protein content in the pollen determines its nutritional quality. Note that in colonies, both protein content and quality influence longevity and performance, and the efficiency of a bee's digestion determines the pollen's nutritional value. We know that pollen from different plant species can vary in both composition and bee digestibility. For example, corn, one of the world's most commonly grown crops, has pollen that's considered poor quality with a protein content of about 15%. So, bees that are raised near crop monocultures like corn could experience poor development. This paper tackles the issue of nutritional stress on pollinator decline. Specifically, it attempts to determine how bees' nutritional needs can be met in monocultural landscapes of low pollen quality, helping to mitigate pollinator decline.
Corn monoculture is prevalent for beekeepers, so researchers chose to examine the common corn crop, which produces a lot of pollen but is poor in protein content. As mentioned previously, the efficiency of a bee's digestion determines pollen nutritional value, and different forms of preparation may make it easier for bees to digest the pollen. This study starts by asking a couple of questions. Can the nutritional value of corn protein be improved by crushing its external pollen wall? What is the effect of bees who consumed crushed versus non-crushed corn pollen grains? Considering specific changes in diet consumption, digestibility, hemolymph protein content, hypopharyngeal gland HPG size, and thorax weight under laboratory conditions. Researchers hypothesized that shattering the corn pollen's exterior wall would modulate diet consumption and digestibility, as well as honeybee worker physiology.
To contextualize our paper, we first observed that protein digestibility is affected by two factors. One, the thickness of the pollen wall, and two, the number of germination pores. So how does pollen actually get digested? Initial hypotheses suggested that osmotic pressure could burst pollen grains, and that no physical degradation of the external wall was necessary to digest pollen. However, subsequent studies in the 80s actually revealed the pollen wall's indigestibility. Keeping this in mind for future experiments, Kralstrom et al.'s 1992 study measured pollen content in the hypopharyngeal glands, or HPG glands, and the gastrointestinal tract. They found that the amount of pollen in the digestive tract was related to the bee's status in the colony. They also found a positive correlation with the amount of pollen consumed and the developmental stage of the HPG glands. Another experiment was done by Rolston and Kane in 2000, where they fed bees pollen diets of varying protein compositions, showing that protein-rich pollen resulted in larger offspring, and emphasizing the impact of food type on body size. Building on all of these insights, our paper explores the interaction of pollen consumption, digestibility, and its effects on bee physiology.
For this study, two different experiments were carried out. The first to test diet digestibility and consumption, and the second to test HPG development and thorax weight. Corn pollen collection for this study was done by placing pollen traps at the entrance of hives to collect corn pollen during harvest season. The corn pollen was then crushed until it became a fine powder with a mortar or pestle. A scanning electron microscope was used to verify that the pollen was sufficiently crushed. The bees used in this study were 3 colonies of A. mellifera carnica treated under similar conditions. These colonies were placed in an incubator, and newly emerged bees from these colonies were randomly selected and assigned to their treatments the next day. In the first experiment, there were 2 different experimental groups that were either fed crushed pollen or non-crushed pollen. Each group had cages containing newly emerged bees set to begin the experimental timeline at day 0. The bees in both experimental groups were fed their respective pollen diets of either crushed or non-crushed and sugar syrup for 10 days. After 10 days, all the bees were only fed sugar syrup for 4 more days. 12-day-old workers were chosen randomly from each of the experimental cages to assess diet digestibility. Their hindguts were extracted and weighed to determine protein concentrations. Protein concentrations were also measured in the corn pollen that they consumed using 3 random samples from the days of corn pollen digestion. The approximate digestibility was then determined using this data through the formula shown on the slide. Then, the total protein content in the hemolymph of 14-day-old bees were quantified using the same formula as above, but instead using their protein concentrations in hemolymph. The second experiment was designed to analyze the effect of crushed pollen on HPG development and thorax weight. For this experiment, the same initial conditions as the first experiment were used, with 2 different experimental groups and 3 cages of newly emerged bees. The bees were fed their corn pollen and sugar diets for the first 10 days. However, after 10 days, they were fed sugar syrup for an additional 6 days. Bees were then chosen at random from each cage at the age of 6 days to measure HPG acini volume. At the end of the experiment, samples of around 10 bees were sampled and dissected from each cage at the age of 16 days to determine the fresh and dry weight of the thorax. The top image displays the cage that the bees in this experiment were held in and the components of their cage used for consumption. This experimental timeline neatly condenses the structure of the 2 experiments that were described.
And without further ado, let's review some of the data and findings from the study. In the first experiment, the researchers found that diet digestibility of crushed corn pollen grains saw a significant increase. This is a result that makes sense, as bees are incapable of digesting pollen grains through the external pollen wall. The authors of the study suggest that grinding poor quality pollen could become a new mechanical method of promoting bee feeding, improving the nutritional value of these otherwise low-protein pollen sources. Additionally, as a result of this increase in digestibility, total hemolymph protein content observed in the bees increased significantly as well. This is relevant in the context of previous research within the field, as hemolymph protein content is usually used to assess the efficacy of protein diets and pollen quality. High protein concentrations in bees have been found to increase lifespan, immunity, and resistance to pathogens. A key discovery in the study is a notable reduction in consumption among bees fed crushed pollen when compared to those on a diet of non-crushed pollen. This paradoxical result, less consumption, yet higher digestibility and hemolymph protein content, aligns with the concept of the geometric framework for nutrition. It suggests that bees, like other animals, strategically adjust their intake to achieve the optimal nutrient balance, emphasizing the complexity of nutritional trade-offs in their diet. In transitioning to the second experiment, it's important to understand why this study focused on hypopharyngeal gland, HPG, and thorax development. The HPG, which is responsible for producing proteins vital for larval growth, was assessed by measuring acini volume. Larger acini indicate more developed glands. Simultaneously, the thorax, which houses the flight muscles, was weighed as a proxy for muscle development. The researchers hypothesized that enhanced flight muscle development might correlate with higher protein intake. And this dual assessment gives us valuable insight into how a crushed pollen diet might not just impact hemolymph protein content, but also crucial aspects of bee development. Acini volume and thorax dry weight did not see a significant increase in this experiment. However, previous literature suggests a positive relationship between the level of pollen consumption and the quality of pollen consumed with the degree of HPG development, so the authors suggest that further research might be required. In conclusion, to address the critical issue of pollinator decline, with a particular focus on the nutritional stressors impacting Western honeybees, this study set out to understand how bee nutritional needs can be met in monocultural landscapes characterized by low-quality pollen. The first experiment of this study yielded intriguing and promising results. Crushed pollen, despite being consumed in lesser quantities, demonstrated higher rates of digestibility, a critical factor in nutrient absorption for bees. Since pollen is the primary protein source for adult bees, and access to high-quality protein sources is essential for colony longevity and performance, this improvement shows initial promise in mitigating bee feeding inefficiencies in monoculture agricultural landscapes. And while the anticipated improved development in bee biology from the second experiment did not manifest significantly, it opens the door to further exploration. Existing literature already shows intricate relationships between pollen consumption, quality, and body development, prompting the need for additional research to unveil more nuanced patterns. Regardless, this study marks a significant step in understanding and addressing the threat of nutritional stress on honeybees. And as we delve deeper into the complexities of bee nutrition, particularly how the effect of crushed corn pollen might affect larval development and colony development, we believe that these insights pave the way for future sustainable practices while fostering the well-being of honeybees and honeybee colonies, contributing to the conservation of these vital pollinators. Thank you.